we discussed our gated, non-gated, and then we discussed ligand gated and voltage gated. What did we say was the purpose of the ligand gated? I've got my protein, and remember, I'm going to draw one, but there will be thousands, and they will only be found at this point. They will not be found anywhere else on the skeletal muscle. So, I'm going to draw one of my ligand gated. It's got a gate. And then somewhere I have attachments for the ligand. What's my ligand? It's what got released by the nerve, the acetylcholine. So the acetylcholine that's gotten released all out here, all right, it's going to fit into the areas on my ligand-gated channel. Is everybody with me? All right, we're talking about muscle contraction, and in order for the muscles to contract, they've got to get a signal from the nerve. Since I'm looking at a skeletal muscle, this means that it's being done by acetylcholine, that neurotransmitter. Acetylcholine, when it attaches to the ligand gated, what does it open? It opens the gate, okay? It's going to open the gate. That channel is now open. What ion did we say was in high amounts on the outside?
Sodium.
These are going to be my sodiums. All right, so these are my positives. And I've got bunches of them, tons, thousands. On the inside of my cell, it's negative. But I also have potassium, which is positive. But what kind of channel was letting potassium, for the most part potassium, just go back and forth, back and forth, back and forth? And it's open, and it's just simply the non-gated channel. So that non-gated protein just simply lets potassium go in and out, helping to keep it negative on the inside. But now that this nerve has sent a signal to this skeletal muscle, and that acetylcholine attached to this ligand, and that gate open, what now rushed through this ligand-gated channel?
Sodium.
Sodium. Sodium now rushed through my ligand-gated channel. What did that do to the negativity on the inside of my cell?
Change the polarity.
I changed it. I depolarized it. And I'm making it more positive. Now, what type of protein do you think might be found next? My voltage gated. So now I'm going to have a voltage gated protein that has a gate over it. So this one, voltage gated, but my polarity now changed. And this is going to affect the voltage gated channels. What do you think happened to the gate? It opened. That gate opened. What rushed in?
Sodium.
Sodiums. Sodiums, once again, rush in. I begin to continue to change my polarity. This will continue all the way down the length of this skeletal muscle fiber. And as that fiber in the muscle does it, as the next one does it, as the next one does it, as the next one, until that whole bundle right there does it, and then the muscle's going to go contract, and then can that muscle stay contracted for a long time? No. I got to make sure this muscle relaxes. Now, that's a whole lot. But inside this plasma membrane, I've taken it from being negative to positive. Does the cell want to stay that way? No, it doesn't. I'm going to need to now balance this membrane. What protein am I going to need to make this stop? My sodium potassium pump. My sodium potassium pump is going to require ATP. All right? Now, because I have changed the inside of this membrane to being more positive, I can't let it stay that way, because when that muscle contracts, the muscle needs to release. So, my ATP powered pump, the sodium potassium pump, with ATP being present. Okay, this is the key here. Okay, so this means that that muscle cell has got a lot going on. One of the things that's happening needs to be generating ATP. So if ATP is present and my sodium potassium pump works, what is going to now happen? Well, and it's going to like repolarize, and to repolarize, I'm going to have to make sodiums go out. This is why it requires energy. Anytime a substance has to move against its concentration gradient, you have to have energy. Sodiums are higher on the outside than they are the inside, and to get them out, the sodium-potassium pump has to push them out. So we have that 3-to-2 ratio. For every 3 potassiums, 2 sodiums go by. So what will happen is potassiums will get to come back in, sodiums get pushed out, and I now make my membrane go back to negative. This is a small part of what happens for a skeletal muscle contraction, but this is what has to occur for it to take place. Those proteins, this is what they're doing. We have to have the ligand gated, that something attaches to, opens the gate, allows the sodium to rush in, which affects the next protein, which is voltage gated. Voltage gated opens, lets more sodium in, continue to depolarize the entire membrane until sodium-potassium pumps balance it back out and take it back to rest. This occurs down the entire muscle fiber. So if we look at that structure, this is the, now remember, only one point for the attachment to the nerve. But throughout that entire fiber, these are located, voltage gated, sodium potassium, voltage gated, sodium potassium, voltage gated, sodium potassium. And it does it the entire way until everything's brought back to rest. Questions? If you understand this concept, you understand this class.
So is it like a domino effect?
It is like a domino effect. In other words, when you get, it's going to almost be like it's in sections where you get this section, this one will get started, this one begins to rest, this one will get started, this one begins to rest, this one gets started, this one begins to rest, and it does it the entire muscle fiber, muscle cell. Now what?
So now I understand about the whole lines and circle thing now.
The whole lines and circles?
Yeah, the membrane.
Oh, the membrane. Now, this is what is taking place for the cells to have their electricity. All right? Because as this is occurring on the skeletal muscle, well, guess what? For the transmission down the nerve fiber, it's almost the exact same thing. It's sending the transmission to release the acetylcholine, to attach to the ligands, to affect the voltage gated, to change my polarization, to get sodium potassium pumped back out, to keep the process going.
So after rest, it's going to do the ligated and the voltage gated all over again, is what you're saying?
Until the transmission stops, or we run out of certain things in the cell, such as sodium or potassium. Now, because it's muscle, it could also be calcium. Okay? Is that not the coolest thing ever?